So we just heard about two different types of things, right? Andrea first talked about the microscope and the importance of developing new kinds of microscopes and improving on the kinds of microscopes that we already have. And Drew was talking about some of the challenges involved in taking the types of data that you get from imaging and creating new sorts of animations and visualizations that really give you insight into how biological mechanisms work. And so my talk, I'm kind of hoping to bring actually those two aspects together in a single example. It's actually emerged from my work. So we're going to specifically talk about certain folding properties of the human genome. We've heard that the genome is very, very long. Just to sum up, to make sure that we're all on the same page here. So a genome is a long set of instructions written in a 4-letter chemical alphabet, which we just saw some beautiful visualizations of. And they are responsible for two different sorts of things. One is to make sure that we kind of can preserve some sort of steady state at this level of cells, at the level of our organs, at the level of the whole organism. And the second is that pretty much all genomes are trying to take over the world by making copies of themselves. And you saw a particularly aggressive example of that in the case of malaria, although probably, I don't know, the rainforest views us as a very aggressive example of that. So this is what genomes are interested in doing. Genomes can be very, very, very long. They're pretty much all generically long. Does the sound seem like it's working okay for people? Or okay, I can tell. So genomes vary a great deal in length. For instance, a virus, its genome is about 10,000 letters long. To give you a sense of that, that's about the length of a chapter in a book. Bacterial, excuse me, that's wrong. It's the length of like a short essay, perhaps. Bacteria have genomes that are about 1 million letters long. So that's about the length of a book, right? The kind of thing that would appear on the bestseller list. After that, these sorts of things get just much, much, much longer. Yeast, which is a very simple organism, has about 10 million letters in its genome. The fly that we saw infecting a human with malaria has about 100 million letters in its genome. So now we're talking about a whole bookshelf. And then an ordinary mammal typically has of order billions of letters in its genome. So for instance, the human genome contains 3 billion letters. So we're talking about, at this point, a good-sized library. I should give a sense, I think though Winston Churchill is said to have reported, who was a voracious reader of books, is said to have read about 5,000 books in his lifetime. So the total number of letters that he read is the length of the human genome. It's also long in a different sense, right? It's not just 3 billion letters long. When you stretch out one of these molecules, it's 2 meters long. So this is something that you sometimes lose sight of, right? In that incredibly folded configuration that Drew was showing, it's very, very compact, very, very, very dense, right? And so it's fitting inside the nucleus of the cell, which is only a few microns wide. But were you to actually stretch it out, your genome is taller than you are. It's very, very long. A basic question is, how does it manage to fold? And there's all kinds of pictures in textbooks about how this works. And I mean, I think this sort of work is very, very heroic. The idea of saying, we're going to take everything that we know and distill it down into a picture of the way things work. The one thing that you sometimes miss in these sorts of artistic renderings is which parts of this picture do we understand very well, and which parts of the picture are we not so sure about. So for instance, if you start at the bottom with the structure of 10 base pairs, right, that looks like a double helix. Pretty well understood. Look at 100 base pairs, 100 letters of the genetic code. Those form this structure known as the nucleosome, which consists of the double helix wound around these chromatin proteins. And actually, you saw those also in Drew's animation. It's also well understood. What Drew's animation didn't show you is this 30 nanometer chromatin fiber, which is, contains, say, 1,000 letters. And then, at the 100,000 letter scale, it didn't show you the extended scaffold associated form, and at the scale of 1,000,000 letters, it didn't show you the condensed, this extended scaffold associated form. At least I didn't notice it. And then at much, much larger scale, you have this metaphase chromosome, which is the way this thing typically looks in pictures you get from microscopes. Well, it's actually good that Drew didn't show you all this stuff, because the truth of the matter is that the stuff his animation showed, right, is the stuff that people actually understand really well. And this whole thing, well, it was very clear how this figure was made, and you can see this figure in every textbook on molecular biology. The artist was told, look, you need to make us a picture showing all the different scales, so you just read all the papers and take your best guess. And it turns out that, when that guess is the double helix at the scale of 10 letters, that's a very, very good guess. When that guess is, that 1,000,000 letters of the human genome form this condensed, extended, scaffold-associated form, that's not a very good guess, because the level of scientific information that's available is actually quite poor at present for this sort of thing. So anyway, there's actually this huge range of scales between about 100 letters and then about 100,000,000 letters. And these ideas about metaphase chromosomes and exactly how they fold, those are actually pretty well understood as well. So, but between about 100 letters and about 100,000,000 letters, we're talking about 6 orders of magnitude, a millionfold scale within which it's quite poorly understood how the human genome folds. A typical thing that people can do right now with an optical microscope, with a light microscope, the traditional type of microscope that's being developed for many, many centuries, is this. You can make particular pieces of the genome light up in different colors. You kind of run out of colors quite quickly. So for instance, here, 2 colors have been used, maybe 5. So you can look at 5 different places in the genome and see where they are under the microscope. Of course, the genome has 3 billion letters, so this doesn't necessarily tell you much about the overarching structure, it just tells you where these 2 positions are. So what I'm going to describe is a technique that we call Hi-C, which you can think of as a very funny sort of microscope. How does the sort of Hi-C microscope work, if you will? So people have, since the advent of the Human Genome Project, gotten very, very good at the following technological trick, which is if you give them a piece of DNA that consists of a whole bunch of letters, they can tell you what the letters are in order, right? So if it's 1,000,000 pieces of, DNA, each of which has 500 letters in it, you can very easily and cheaply read off what all those 1,500,000 letter sequences are. This is a very, very, very powerful enabling technology, DNA sequencing, and every day this gets cheaper and faster and better, and it's really undergoing an explosion, very, very much like the explosion in, for instance, desktop computing, right, that took place in the 80s and the 90s and 2000s, and that is in many respects continuing to take place today, where computation is easier, cheaper, and faster. So the idea that we had is, can we use a DNA sequencer, right, a machine that takes little stretches of DNA and reads off what the sequences are, and can we convert that somehow into a microscope for reading off how the human genome folds? And here is basically the trick. So what we're going to try to do is to try to solve the following problem, which is whenever 2 pieces of DNA bump into each other in the genome, right? If the genome is folded up in some sort of configuration, and as we saw in Drew's animations, these things are constantly undergoing all kinds of vibrations and fluctuations. So things bump into each other, right? When they bump into each other, we want to know that that happens. What we want to do is really identify pieces of the genome that have bumped into each other and read them off. And the basic way that this technique works, and I'm not going to take you through all of the details, is that you start with 2 pieces of DNA that happened to bump into each other. You cause those pieces of DNA to fuse and form a loop that looks kind of like this. And you see there's these interesting parts of the loop where, one piece of DNA and another piece of DNA have fused. So you read off, you excise those pieces that have fused, and you read off all of those little fusion products. So the genome contains a blue sequence, maybe it's somewhere in chromosome 1. It contains a red sequence, maybe it's somewhere in chromosome 3. But now we have this funny blue-red fusion sequence. That isn't a natural genetic sequence, right? It doesn't exist anywhere in the genome. It's just the fusion of a piece of chromosome 1 and a piece of chromosome 3. Now, if someone were to read off those bases to me and I were to refer to a database of the human genome, I could figure out, oh, part of that's on chromosome 1, part of that's on chromosome 3. And if I knew that this experiment had taken place and had produced this fusion product, I would know that such and such a part of chromosome 1 and such a part of chromosome 3 had bumped into one another. So that's good. There's a whole bunch of disclaimers about all the ways in which this does and doesn't work. And there's many good reasons for skepticism. In fact, actually, when the original microscopes were developed, folks were skeptical about them for like over a century, for a number of centuries. People actually thought that if you can't see something with your naked eye, it might not really exist. And it wasn't actually until a very beautiful form of microscopy was developed, I think in the 14th century, in which what you would do is you would take the image and instead of having like a traditional microscope where it went through an optical lens, you actually had this object that you wanted to magnify, and then its image got reflected off a very curved mirror. And so there was no lens interposing between yourself and the thing that you were trying to image, and that convinced a lot of people that this microscope thing might not be total BS. Anyway, so that's the nature of things, right? When people develop new imaging techniques, there's always all kinds of disclaimers and reasons for skepticism and things like that. And we're trying to develop this into a powerful technique, and so far we've made some progress. So let me give you a sense of what the kinds of data that a Hi-C experiment produces are. So along this sort of X and Y axis, you have positions in the human genome. And this is the type of data that's produced by the experiment. So you can see it's basically telling us the number of times that a particular position in the human genome bumps into a particular different position in the human genome. There are the 23 chromosomes here along this diagonal. So these are bumps within chromosome 1, bumps within chromosome 2, bumps within chromosome 3, et cetera, et cetera, et cetera. So they're very, very massive data types that require visualization. Let's zoom in on one of them. Gives you a bit of a sense. This is just chromosome 8, and these are the regions in chromosome 8 and how often they bump into one another. And just to give you a sense, let's suppose I'm interested in some gene, such and such a position. This is actually very large for a gene, so that wouldn't be the case. But suppose I'm interested in a gene that's in region A, and I want to understand something about region A. So how do I interpret this experiment? Well, I would look and I would see first, region A bumps into itself a lot, which makes sense. You bump a lot into your next door neighbors and the things that are close to you in 1D. And so this is why you see this incredibly intense diagonal in these experiments, because all regions bump into themselves a lot. But you see relatively fewer interactions with, say, region B, and then still fewer interactions with region C. And more generally, you can see that there's these complex patterns here. There's these funny polka dots, there's this interesting starburst structure. So there's all kinds of interesting features in these maps that we can strive to interpret. And there's a very strong correlation, actually, between numbers of times that things bump into each other and their physical distance as measured by a microscope. So one way of thinking about this experiment is that it is this experiment, which you can use to measure the distance between two sites in the human genome, but you're doing tens of millions or even billions of such experiments all at once by measuring the frequency of bumping instead of average 3D distance. Anyway, I can tell you a lot of things that folks have learned about the structure of the nucleus using this sort of thing. It's a new technique that people are excited about, and there's certainly a number of interesting things that you can observe. But what I really want to do is, people have observed these sorts of chromosome territories, which are actually Drew's animation. They've observed, this is actually something I reported in 2009, segregation between active and inactive pieces of components of the human genome. So when genes turn on and off, they're actually being moved in 3D. But what I want to focus on is a model that we kind of really sort of introduced, but sort of reintroduced on the basis of our 2009 initial data of this type for how the human genome folds at the scale of a single megabase. And I want to just step back and do a tiny bit of philosophy before we jump into this. So a megabase is 1 million genetic letters, right? It's information, but it's also a polymer because it's the same chemical over and over. So there's two different types of ways we might expect to think about it. If you think about it as a polymer, then that immediately suggests to you that, well, there's a whole branch of physics called polymer physics, and polymer physics should be able to tell you something about how the genome folds. about it as information, right? We know about information. We know how to deal with information, right? It's a lot of information, so it should be like a library. And a good library is compact. All the books are in one place. Organized, you should be able to find what you're looking for. And highly accessible. When you find what you're looking for, you can just pull it off the shelf and leaf through it. One last preliminary thing before we kind of dive into the synthesis of all these things. So if you have any curve, right, this is just a property of curves. It has nothing to do with genomes or anything like that. It has what we might call a contact probability function. So you can see this curve has been drawn from blue to red. And you can see that sometimes things bump into one another. Right, and this is for a short distance bump, and you can tell it's very similar colors. This is a long distance bump. Right, this is a distance 3 bump, distance 213 bump. And actually, in these pictures that I'm going to show you, can tell how far, whether it's a long distance or short distance bump in the curve, because you'll see very similar, colors for short distance bumps and very different colors for long distance bumps. And you can make a table of this, or you can make a chart showing this. Many different ways represent the contact probability function of a curve. This is the contact probability function averaged over the human genome. And there's actually a very striking statistical regime here where the contact probability, the likelihood that two things are bumping, scales as the reciprocal of their one-dimensional distance. And this is a very funny sort of behavior that we observe. Right? So if things are twice as far apart in this regime, they're half as likely to form contact. What could possibly explain that? Remember, we're trying to figure out what the structure of Megabase is. It should both be a good library, but also we have some ideas. We think that this kind of thing maybe is emerging from the polymer physics. There's actually a very classic model for how long polymers fold up, called the equilibrium globule. This is a picture of one. It's a highly compact structure, so you can understand how it folds so densely, the way that we saw in Drew's animation. But it's a big mess. Right, there's very little correlation between 1D and 3D structure. It's actually a highly knotted structure. One of the big problems with it is that it doesn't actually give this statistical scaling that we saw, this reciprocal relationship. Right, it gives a related relationship, which is called a power law with an exponent of -3/2. A reciprocal relationship, just to get the terminology out there, is basically a power law with slope with -1. This other structure that nobody really talks about, and the equilibrium globule is sort of the classic structure. So this is part of what got Ford his Nobel Prize in the 70s. It's really the classic picture of how polymer condenses. This paper, written though in the late 80s, that says, well, actually, when a very long polymer condenses, it might condense into this different structure called a fractal globule. And if you just look at a picture of this, it's highly dense. You can see that, but it's also highly organized, right? There's a very strong correlation between where things are in 1D and where they are in 3D. And the theory actually suggests that it should be completely unknotted. And crucially, I was able to argue that it ought to give you a scaling of -1. This reciprocal relationship is true for a fractal globule. When things are twice as far apart, they're half as likely to be bumping into one another. It's actually a very intuitive idea. So there's this concept, basically, what they suggested is that initially something folds up into a fractal globule, and then after a very, very long time, it transitions into the equilibrium globule. So they weren't arguing with this idea of eventually a polymer will form that. They just said it will take a very long time, and for a polymer like the human genome, it could take millennia to make this transition. And that's actually a very intuitive idea. I don't know if many of you are familiar with this particular substance. I don't know, in the US, this is like what graduate students eat for like 10 years of their lives. But I don't know if like here, people ever eat this. Do any of you guys recognize this? Yeah. Perfect, wonderful. I'm glad that some culinary traditions are universal. I was trying to, like make an argument for why this is somehow similar to mutter paneer last night, but I got nowhere on that. So this is ramen noodles. It's great. This actually is, you can purchase this for 25 cents. So I guess about 10 rupees at the pharmacy right across the street from where I live. So it's, within the U.S. there's not so much money. So anyway, so you can buy this. It's quite cheap. Does anyone actually know roughly what the length of the noodles it's a 4" by 4" by 1" package. Does anyone actually have a sense of the total length of the noodles in this package? Any guesses? I mean, you guys are, you know, this is IIT Mumbai. You guys should be able to figure out how much noodles you're buying per rupee. Any guesses as to the length of the noodles? 50 meters. That's actually great. That's pretty much dead on. You're buying about 50 meters worth of noodles, which is quite impressive. I think people never get that right. Most people give dramatic underestimates for some reason. But yeah, it's about 50 meters worth of noodles that you're purchasing, which is actually very simple to figure out. I was trying to figure this out, and I decided what I would do is very simple. I'd just boil these up and then go to a nearby parking lot and lay them all out instead, and then get out a tape measure. But then I had a crucial scientific insight, which is that if someone had ever done this, they would surely have written a blog about it. And it turns out that they had, and they did. And so there's really outstanding statistics available for the whole length distribution of ramen noodles and all of that. Now the critical thing about ramen noodles, right, is it's very, very, very dense. But it's folded up in a very interesting way, right? Which is that it's totally unknotted. Like, so if you put it into the water and then you wait a couple of minutes and you stick a fork in it, it hasn't cooked terribly much. You pull out a couple of noodles, but you've actually not disrupted the structure. Then you've got to put it back, right? So you basically don't disrupt the structure at all when it's in its original configuration. But now if you actually wait and let it cook, then eventually it transitions, right? It undergoes this state transition to your cooked noodles. Now the cooked noodles are equally dense, more or less, to the original configuration, but they've undergone a topological transformation, which is that they're now highly knotted. Right? You know this because if you stick a fork in, you get this whole big problem. Right? So this is really experimental polymer physics. So this is pretty much the transition from the fractal to the equilibrium globule, on your stove. So it's neat that the fractal globule is consistent with the high CA. And I want to just, why is that interesting? Why is that interesting to us biologically? Well, I can show you these beautiful pictures, but in fact, this is one created by Mary Huntley and the group last year. This kind of thing wasn't available when we originally did this work. In fact, this was sort of the state of the art of how you visualized fractal globule. This is a drawing made by Shura Grossberg, who originally suggested this structure. In fact, it wasn't made by him, it was made by his 12-year-old daughter in 1988. So this is the state of the art. It's actually pretty good for what it's trying to do, and better than I can draw. But there are very big advantages of the kinds of physics-based visualization systems that exist right now in terms of their ability to give you insight into the structures that you're looking at. So we're able to actually fold these fractal globules up from scratch. So here's an animation from a couple of people in my group. You can see this polymer folds up. It forms these little bundles as it spontaneously folds in a pore solvent. Those bundles start to collide into each other, forming bundles of bundles. And then those bundles of bundles sort of fold into each other, forming bundles of bundles of bundles of bundles, globule of globule of globule of globules, hence the term fractal globules. So we can create these sorts of things, and then we can do little experiments with them. So for instance, I can check if it's topologically knotted or not using the following sort of tricks. So those of you in the audience who have math backgrounds know that something is, the definition of whether something is knotted is almost defined for a closed curve. So we have to add an ear, since that was an open curve. We have this little ear to check whether it's knotted. So now we're going to track it down. And the whole thing, so this is basically just applying essentially a spring force along the length of the curve. And you can see it contracts down very nicely because it's completely unknotted. If I were to do the same experiment with an equilibrium globule, you don't get anything nearly as nice. See, initially it's doing very, very well, but eventually you realize it's a highly knotted structure, so it cannot continue to contract. Now this knottedness is not just a mathematically interesting property. This is actually pretty significant biologically, because what Drew showed you in his animations is this configuration where in particular in those animations where he was showing you how the DNA replicates, he was showing you this DNA as a very extended state on which all these polymers can land. But how does the DNA stretch out that way? Because if all the DNA was that stretched out, again, this thing would be multiple meters long, so it wouldn't fit. So you need some mechanism for being able to pull out a particular piece of the human genome and kind of stretch it out while not disrupting the rest of that dense stuff. And what's really nice about the fractal globule is that it enables just this. So here we're just pulling on this yellow region, and you can see this part will stretch and open up. The rest of it is completely undisturbed. So having this kind of incredibly dense, unknotted structure is actually really, really valuable for allowing you to store all this information in an incredibly compact way, but also pull some of it apart when you need to copy it in the ways that Drew has shown. Anyway, I want to thank all my collaborators on this, members of my group, as well as Nico Van Berk from the Filmates, initially developed the technology, and all of our funders. And I want to point out, Tim, starting My Lab is Moving, what's missing is potentially you. There are openings available. I know this is IIT Bombay, or IIT Mumbai, it's loaded with smart people, so feel free to be in touch with me if you find the kinds of things we do interesting. Anyway, thank you guys so much for your time.